All right. Thank you all for joining today. As a reminder, the public webinar series is designed to educate attendees on the scientific ecosystem, and every month we'll have either a representation from the Data Coordinating Center that are available and new developments from the centers that support them. Okay. So today we're going to walk through a few scientific updates from the KC, going through some upcoming events throughout the CFDE, and we'll hear from our featured speakers. So just a reminder, if you were unable to attend any of the webinars from the past, all of the recordings are available on our YouTube. This is a link to our YouTube specifically, and you can go back and look through the various ones there through the playlists. Just a reminder, as the KC, we'd love to create use cases that are specific to your research, specifically with anything that you would use for the data sets that might be within the various DCCs. Just a reminder, this is our landing page and our site address is here. So with that, if you have any feedback, questions, or would like to be added to our listserv, please feel free to email us at help@cfde.knowledge.org. We'll be able to answer any questions that you may have as well. Just going over some of the events that we've had. On the 24th and 25th this past week, the KC and DRC were graciously invited to participate in the NIH Common Fund's Community Engagement Workshop. This is a great opportunity for all of us to meet in person, which is always great to bounce ideas off of each other. And essentially, at this session, we reviewed barriers that currently exist into using data that's available within the ecosystem, and also learned about various outreach, training, and support that can be provided within the community. It was great to hear again from the various perspectives, not only from the CFDE, but Common Fund as a whole. So thank you again for having us participate in this event. Okay, so later this year, we're going to attend a large conference called the American Society of Human Genetics. This will be held November 5th through 9th at the Colorado Convention Center in Denver. So we are excited to announce that in addition to our booth, we'll also be hosting an ancillary session that will take place on the 6th of November, and this will be from 11:45 to 1:15 p.m. in the Convention Center. We'll have some more details for you later down the road as we plan this further out, but please feel free to have this bookmarked and ready if you will be attending this conference. We're excited to see everyone, and we hope this is another opportunity to have various DCCs and centers come together to not only share information about the CFDE as a whole, but our resources and learn more from each other. Some other events that I'd like to make aware to the group within the next few months. The first is the Independent Study in Biomedical Data Science from IDG, which is the 27th. That's coming up this week. The Spark Codathon, the signups are still available until the 27th, which is again this week. So you want to make sure if you're going to be participating that you sign up by then. And the Kids First CFDE Pediatric Power Up will take place in August. All of this information will be available on Twitter as well and through our various resources if you're interested in learning more. So today we're going to have a tour of the Human Reference Atlas v.2.1, and we'll have Katie and Bruce who will be sharing their story, and so we thank them for joining us today. Before we go into this, HuBMAP and Senet, who are the two representatives here speaking on the HRA, there are two resources within the Common Fund data ecosystem that serve as data coordinating centers, and this shows you how they fit in a little bit here. And as I mentioned, the Human Reference Atlas is the main topic of discussion today. The Atlas is a multi-scale, multi-model, 3D atlas of anatomical structures. And this is the landing page, just to give you a little show as to what we will be talking about today. And then before I pass it off, I'll hand it over to Noelle, who will give us a little bit of framing with the KC.
Thanks, Mackenzie. Actually, that was a great intro, and I think I'm just more excited. I just wanted to share some excitement about having Katie and Bruce online with us today, because some of the work that the HuBMAP is doing and Senet are doing more broadly are incredibly useful to so many resources that I'm involved in as the Knowledge Center, as PF and Knowledge Center, but also more importantly, as the broader research community is involved in generating these types of data types and visualizing them specifically for diseases. And I think the HRA and the HuBMAP are really leading the charge in terms of creating standards, easy visualizations, representation of this data that makes it useful. And I thought that was really, I'm really excited to have them come, because I think I'm hoping that folks from our disease communities will join the call today to sort of hear about this. But with my hat on as the Knowledge Center, we're building a resource which is very early stage, and the mission of the CFDE is really to enable better use of the common fund data sets. And so our thinking behind the webinar is to literally just share these resources with you all and start to get feedback really about how you may have used them in your setting, or how they could be extended with the data that they have to answer a different scientific question that they may not be addressing, because they're really in many cases, data generating and data representing warehouses. But we want to make the goal is really to enable these common fund resources to enable discovery and accelerate our understanding of health and human disease, right? So our role as a knowledge center is to help put that forth. And the landscape of data is incredible in the resource, right? This is something that, the first pilot phase did to generate for all the assays and files and data sets. And it just gives you a sense of the landscape. But each of these data set DCCs are their own rich resources you're going to learn around today, these two resources. And they have their own consortium surrounding them. So it's a pretty large remit for us, I think, to try and figure out how to represent knowledge from them. Because moreover, like for an example for HuBMAP, which Katie and Bruce are going to echo better than I can, there's a consortium on top of this that is generating the data, contributing to the resource, but also making insights and publishing on those. But those publications also have analyses that could be made accessible more readily. In fact, they had so much from this consortium that they have an entire issue of Nature devoted to them. This is amazing stuff. This is the type of thing that really is useful to the broader field at large. So I hope we hear about that a little bit today. But our job as a knowledge center is to help sort of bring you, if you are an experimental biologist, if you're a human geneticist, if you're a clinician, if you're just curious as a scientist in general, bring you to the various resources in the Common Fund resource and help you understand what are the scientific questions you can ask, what are the data types, what are the various tools that already exist, and sort of bring that to you in a way that's a little bit more straightforward and simple, so that if you are sort of like really interested in digging down, you can. And we're very lucky. We're also funded by the yin to our yang. Our co-team is the CFD work branch, and their job is to bring all the data together and the metadata and the standards. And together we form this sort of infrastructure that helps sort of contextualize all that is Common Fund. And the way we think about doing this is really the 80-20 rule, and the idea that, we want to bring this to you. Those of you who like lobster, I love this reference. Hopefully it's useful to you. We want to bring this presented to you on a plate, already prepared for you because you love lobster. But we want to make sure that, given the fact that the CFD represents a whole host of resources that actually have the raw data processed and standardized and presented with all the metadata, we want to enable it. So if you actually do want to go down deep and actually do all the work to analyze that data that they have on your own, we present both to you. So with that, sort of like the KC is meant to be a front end for all of that. And over time, we hope to build sort of an entry point that presents things in the form of contexts and allows you to navigate based on things you might choose to follow down workflows or paths, with all of the DCCs being infused into that research journey, if you will. So for example, if you pick a different, a particular journey, you determine for HuBMAP what data types they have, what entities they search. And this is just the beginning of some navigational tools. But concurrently with our ability to build a software platform, we want to bring all these resources to the community. So that's sort of like the setup from two angles. And I want to sort of have these part of our conversation more readily. So you, as folks who are using these resources, feel free to contact with us with ideas or things you've done to use these resources in your own setting, because I think that helps us understand how these resources are used more at large. So with that framing, I'm going to turn it over to Katie and Bruce to really talk about HuBMAP and CNET.
Thank you so much for the nice framing and the nice introduction. And thanks for having us. It's very exciting to be back at CFDE. I have attended a number of in-person meetings, and it's always very exciting to see what other teams have been doing. And each of these consortia is like 20 to 200 people, right? They start small, but then they ramp up to production phases. So HuBMAP now has 350 researchers. CENNET is a little smaller, but also ramping up. And of course, it's not just those two consortia which are benefiting and contributing to the Human Reference Atlas, but 20 plus consortia and efforts. And all of this is also supported by the Human Cell Atlas. So it's just super exciting to see so many experts come together to make something that's much more than the sum of parts. So this is a presentation by Bruce and myself. We will be team tagging. So I will do a few slides and then Bruce does the majority of the slides. That's why he's kindly running the slides. And it's a true pleasure to present to you here from Bloomington, Indiana. I lead one of the two mapping cores inside of HuBMAP and CENNET. We are responsible for making an atlas, for creating a common coordinate framework, a latitude-longitude system of the healthy human body. And that's non-trivial because it has to be multi-scale and has to have a lot of other features we care about. And data is still expensive. That's just what it is. So let us guide you through what exists right now. And again, this is work by many experts, not just the two of us. So on the next slide, you see some of the collaborators. Again, it's many more consortia than you even see listed here. Many subject matter domain experts, which with their ORCID IDs are now associated with the Human Reference Atlas forever because they're published every 6 months with all the digital objects that make up the Human Reference Atlas. It's funded mostly by NIH and CIFAR, but also some smaller other pots of money. And again, with much support by the Human Cell Atlas, which has been marvelous in bringing everybody from around the globe together to work on this massive challenge together. On the next slide, you will see a map of the US, but also with some teams being in Europe. And this is just HuBMAP. So imagine there are 20 of these consortia and just coordinating all the efforts and getting all the code pieces to work with each other and having people agree on metadata standards, non-trivial. In HuBMAP, we have tissue mapping centers, TMCs. We have technology development centers and others. And then we have the Hive, which is the technical core. And again, Bruce and myself, we are part of the mapping centers. There are also tools components. And there is the Pittsburgh Supercomputing Center, which is doing the community engagement and providing cloud infrastructures and plug-and-play architectures that ultimately help us get all these software pieces to interoperate with each other. So on the next slide, you see the history. For those of you which are not familiar with HuBMAP, I also posted a link to the HuBMAP data portal in case you want to check that out, and also the HIA portal. But HuBMAP started in 2018, started actually earlier with white paper efforts and other consortia gatherings that ramped up to even having a call for proposals for HuBMAP. And the ultimate goal is to create a map, to create an atlas, to create a common coordinate framework. So it's not just creating data which are high quality and are made available to the community, but really is creating an atlas that you can cross-search across scales, but also across organs and teams and technologies. We are very much focused on creating a common coordinate framework, as you will see, and making it scale, because just cells by themselves typically can't deliver the function that's needed to keep us all alive or to keep other species alive. Oftentimes it's teams of cells that have to productively work together. In 2024, we published the Human Reference Atlas version 2.1. In fact, it just came out in June. And then for Christmas, you get the next release. And so we are in production phase, and it's all fast and furious and exciting. On the next slide, I think that goes over to Bruce now.
Yeah. Okay. Yep. So during the presentation, I'm going to try to give you the high-level overview with the HRA, and then we'll be diving into the individual pieces and parts. You'll see that it's broken down into a series of different layers and pieces that all work together to make this atlas. So at a high level, Human Reference Atlas, it defines the space and shape, the three-dimensional space and shape of anatomical structures, the cell types and biomarkers that make up those anatomical structures. And they are validated and represented using ontologies, primarily Uberon, FMA, cell ontology, and HGNC for biomarkers. We define how new data sets can be mapped to the HRA, both spatially using the common coordinate framework, but also via ontologies, and even data that's experimental data that's been processed. Say with Azimuth, for instance, we're able to map those individual cell annotations to the HRA via ontologies again. So the HRA, it's authoritative, and it's every digital object as part of the core HRA is DOI'd. It's computable, and we have authors and reviewers for every digital object to approve them as it goes through. It's computable. It's published as linked open data, 5-star linked open data for very easy to access, fair data. And it's open. All of our data and code, all of it's available in permissive open source. And then it's continually evolving as we're, the body contains trillions of cells, and it's a very complicated beast, and we are continuing to evolve our atlas so that it can represent more and more of the human body. So we break it down. So with HRA, we sort of, at a conceptual level, we break down the body into organs that then consist of these functional tissue units like the renal corpuscle. And the functional tissue unit, would say, is like the smallest set of cells that perform some function as part of an organ. And then we go down to cells, and then biomarkers, genes, proteins, and the like. So that sort of is sort of a conceptual level. And then in the core atlas, we define anatomical structures, cell types, and biomarkers in these ASCT plus B tables. The 3D reference organs provide 3D models of the anatomical structure. The 2D FDUs provide 2D illustrations of these functional tissue units. HuBMAPs provide a algorithm, a method by which you can process data and more or less be able to annotate your experimental data with cell types and biomarkers from these HuBMAPs. And we are modeling the vascular geometry, and a new digital object type that's added in 2.1 are these cell type annotations. So we have multiple tools, Azimuth, Cell Typist, POP-V, and it will expand over time, but it allows us to say, if you've used Azimuth and use this model that you've trained with Azimuth, your data can be automatically annotated in a way that it can be ingested or brought into the atlas. And so that provides our core. So everything in red has been reviewed by reviewers and signed off by subject matter experts. And then, so on top of the core, we then have some new things come through. So experimental data, like being able to take your data and map it on to the HRA. Being able to, that's the experimental data. HRA POP then can dive into your data and annotate those cells with these cell summaries, and do some other interesting things. And HRA Lit also builds on top of the atlas, providing literature from primarily from PubMed, but may expand over time, literature associated or useful for the atlas. So that's the overall architecture, and we'll dive into the individual pieces and parts, but just wanted to give a high level overview with that. And then, so we'll now talk about user stories, which is an integral part of the HRA.
Right, so user stories are needed because the atlas will ultimately serve many users and many functions, but we need initial user studies to just guide the initial development. And so here you see that there are some user stories that are centered around construction. So they facilitate the construction of anatomical structures and biomarkers and cell types tables, but also of 2D and 3D reference organs. And you see that others are supporting usage. So here you are actually using the initial atlas, which is still in draft format, to serve first initial user stories. And 2 of the user stories guide sustainability, because we would like this atlas to be widely adopted and sustained by many. So in the table that you see on the next slide, you see all of those. And so you always have a feature, which is also the name. You have a user role. What are the user groups for which this is intended? You have the activities that the users want to complete. And you have the value added, the return on investment after having done a lot of data wrangling, a lot of code development and user interface design. What do you actually get to see? And so in order to get to these 7 user stories, we did very many interviews with Atlas architects, 30, and not just those from HuBMAP, but also across the NIH ecosystem and also Human Cell Atlas, and several also in Asia and other continents, because we wanted to have a more global view of what this atlas could look like and how it might be used. And so we also, in addition, interviewed 6 programmers, because ultimately all of this has to be feasible, and programmers are smart. They might identify cool new things of how this atlas could be constructed that maybe some of the biologists don't yet see, because they're not so much on the technical side. So this table can also be found in a recent preprint, and I think Andy just put that in the chat. We would love to have your comments on it. If you see other user stories that you would like to see included, please reach out to us. On the next slide, you see that we, in order to serve these user stories, we need to agree on names for anatomical structures, for functional tissue units, but also for cell types and even cell states. There's progress on that end as well, but also agree on biomarkers that are typically used to characterize these cell types, and to have agreement on which cell types are commonly found in which functional tissue units or which anatomical structures. So that's really what the ASCT+B tables cover. But this naming is non-trivial, because many of the existing ontologies don't yet cover everything that's needed to represent the healthy human body. And so we are working closely with ontology curators and engineers and owners to add relevant new cell types and new anatomical structure names into existing ontologies, such as cell ontology and Uberon and FMA. On the next slide, you see the many different user groups that we are serving, and they all speak different language. They have different cultures. They have different incentive systems to be part of it. Non-trivial, but we have a monthly working group, the ASCT+B working group, which brings us all together so that we can develop a unified language. And we by now had 54 meetings, so this has been going on for a while now. And if you're interested to join that working group, it's completely open. We can give you a survey link. I think Andy can put that in, and if you fill out the form, then you basically gain access to all the recordings, all the slides, but you also get invites to future meetings. So on the next slide, you see what the Human Reference Atlas is trying to do. It tries to capture healthy tissue in support for comparisons with disease, very much like the Human Genome Project tried to get us a sequence of all the human genomes, so genes, so that you can then take snippets and match them against it. So here, similarly, you try to record cell type populations for the entire human body, and then giving a new tissue with a known cell type population, you can then try to figure out where it came from. So that's use case number 1. But the other next use case is that you cut out a tissue from, let's say, via biopsy from the kidney, and you can predict what kind of cell type population you should see if it's a healthy kidney, even before you run any kind of single cell analysis. So that's really 2 core use cases that we have identified and that we are working on. Ultimately, we also want user interfaces so that many can use that data. Not everybody can run a Sparkle query or even a relational database query or use an API. So I think these visual interfaces are very important. And then, however, we also want to interconnect all the programmers that actually want to use the Atlas programmatically and also want to ultimately run machine learning algorithms over it, because I believe, and I think many of us believe, that this mapping exercise cannot be done with human intelligence alone. We really need to combine human and machine intelligence to map our human body at single cell resolution across scales. And we have a lot of open data and open code, many reproducible workflows and lightweight user interface component linked from the HRA portal. So you're very welcome to benefit from this, but also please do let us know if you miss anything, and we can always run custom queries for you. And it's not yet ready for clinical practice. There is no single clinical trial that has been using the Human Reference Atlas. It's just too early. On the next slide, there's a tour of the HRA, and I think that's Bruce again.
Okay, so and I'll quickly give that back over to you, Kadi, because at this point, what we're going to do is go through each of the pieces of the HRA and talk about these. So each cell that you see here, each little square is basically going to be a digital object type. And I'll get into some of the technical details as we go along, but these are individual pieces of components of information that we use to make the larger atlas. So, yeah.
So on the next slide, you see one of those ASCT+B tables laid out as a graph. So on the left-hand side, you have anatomical structures that make up kidney, not all of them. There are many more. That's a subset here. As you see, they are linked via part-of relationships. So for instance, the glomerular capillary endothelium is part of the glomerulus vasculature. It's part of the renal corpuscle, part of the cortex of the kidney, part of the kidney, part of the human body, ultimately. And then in blue here, you have cell types, and again, just a subset of all of the cells that you would find, and they are connected to certain anatomical structures where they are commonly located in. And you also have biomarkers that are commonly used to characterize cell types. And it took not forever, but a long time to have experts agree on these relationships, also agree on names for anatomical structures and cell types and biomarkers. And then again, on our end, we are working with ontology experts to then update ontology so that they reflect healthy human bodies. On the next slide, you see that we also have 3D representations of major anatomical structures, more than 1,000 of those. And we also now have 2D illustrations and hopefully soon 3D representations of functional tissue units, such as here the renal corpuscle from the kidney. And those are used to, again, agree on where certain cell types are, what shape, size, and function they have, and how biomarkers are used to characterize them. And I think it really needs to have this yin-yang of terminology, agreement on anatomical structure, cell type, biomarker terms, and their spatiality, because everything in the human body happens in 3D, and it matters where it is and who is next to it and who is in contact or not, and who is close by to get certain types of signaling to hear. So on the next slide, you have a zoom into the heart. Some of you might be heart experts, and you might see some of your favorite anatomical structures or cell types or biomarkers in here. This is an interactive data visualization, which anyone can access for 31 different organs now. So the ASCT+B reporter allows experts to check the relationships that they have created via tables, but it also helps them communicate to each other why certain linkages are missing or should be added. And this is very valuable ultimately also for creating so-called HuBMAPs, validated antibody panels, that then are used to identify certain cell types using immunohistochemistry approaches. On the next slide, you see the same view, but for kidney. Again, here now you see more anatomical structures, cell types, and biomarkers. And on the next slide, you have one more organ, which is the lung, near and dear to all of our hearts, because without oxygen, we can't do much. And here again, you see that there are very different anatomical structures, but also cell types and biomarkers. And then, of course, there are some cell types and biomarkers that exist across organs, and those are especially interesting for understanding what happens, for instance, to immune cells across organs, or how is the vasculature connected to certain other cell types. And so we have some interfaces for this later. On the next slide, you see again the overview. So we are now going to zoom into these 3D reference organs. On the next slide, you see the anatomical structures, the organs, but each of the organs actually has an inner structure, and there's a really neat Twitter contest going on right now, which Andy is organizing. So you could actually win something. So there are prizes. So Andy, if you want to point everybody to that, I think it's a really neat exercise to see what you see inside of these anatomical structures. So for instance, one of the ones that already run is this visualization. I don't know if you can see this. And so you had to guess what inner anatomical structures exist across these 65 human organs. On the next slide, you see that these organs are then used to register tissue blocks, either derived via biopsies, or in some cases, the tissue mapping centers have access to entire organs, and then they cut out larger blocks of tissue. But they all need to be registered back so that we know the position, the orientation, and the size of these tissue blocks. Not always easy, because now you have very large organs and potentially teeny tiny biopsies and few landmarks only, but we are trying to make this as easy as possible for those which have to do the registration. On the next slide, you see we are going to now zoom into the 2D functional tissue unit illustrations. And so here you see the 23 different functional tissue units that all have a very well defined function. And oftentimes you might not realize the size differences between those. We also now have some hierarchical nested FTUs, such as the nephron tubule here on the right, which actually has a number of sub-functional tissue units. And they are quite beautiful, so you can zoom into them. I think on the next slides you have a sequence of them. Bruce, maybe you can just blend through those. And later on, you will see them in action, because you can actually hover over them to see what kind of cell types exist and what biomarker expression levels are normal for any of these cells, which are captured here in these 2D functional tissue unit illustrations. And as you see, they all have a scale bar, they all have names, and they can also be used as lightweight atlas components. And I think Bruce will demonstrate that later. On the next slide, we are now going to move into organ mapping antibody panels. So again, those are used for Codex or CellDive or other spatially explicit approaches that use validated antibodies, and it takes a lot of time and money to arrive at panels that really work. And so there is an entire HuBMAP framework and a Nature paper that maybe Andy can put into the chat, because we would really like other teams to benefit from all that work that HuBMAP and many other teams have completed, where they have gone through possibilities of what providers have, which kind of antibodies that actually reliably work and work together to identify healthy human cells across different tissues and different preservation methods and different technologies. And so maybe reading the paper gets you the most information on this, but this is a really, really important contribution that HuBMAP is making. On the next slide, you have OMAPs in the same format that you already saw for the AST+B tables, because here also you have certain biomarkers, here now proteins, which are used to characterize certain cell types, and then the cell types are located in certain anatomical structures. So you have this for kidney, and then on the next slide you have it for lung, and you might even have one more, I don't know.
I think we had 2 kidney and 1 lung, but that's I think that's it. That's all right on this one.
So kidney has different assay types, and therefore you have different OMAPs then. On the next slide, you see vascular geometry, very near and dear to our heart, because we believe we could possibly construct in a latitude-longitude system using the vasculature and creating a common coordinate framework this way. And so you see that the aorta coming out of the heart then goes into different cycles throughout the human body, feeding different organs, sometimes going through multiple organs before it comes back to the heart. And it goes across scales, and it feeds nearly every single cell in the human body, not quite all of them, but nearly everyone. And so we are interested in collaborating with teams that work on vasculature data and are interested to help us understand how many vessels are connected in which way, with which branching angles, which diameters of vessels, and what biomarkers can be found where. And so on the next slide, you have a video that kind of explains the concept of the vasculature common coordinate framework. So let's play that.
The Human Reference Atlas, or HRA, is a major international project bringing together over 1,000 scientists to build a map of all the cells in the human body. But how can we describe the location of something as small as a cell within the whole body? Thinking about how a package is shipped to a building might give us a clue. Although a building has latitude and longitude coordinates, the label lists a street address, which is the position along the road nearest to the building. In humans, blood vessels serve a similar purpose as roads, forming a connected set of pathways throughout the body to deliver oxygen and nutrients to cells. With a roadmap of the blood vasculature, we can assign each cell an address relative to the nearest vessel. This is the concept of the Vascular Common Coordinate Framework, or VCCF. Imagine driving down an interstate highway, which we can compare to one of your red blood cells coursing through a major artery. You exit into a city along a large avenue that branches into a network of smaller streets, which pass through different neighborhoods, just like your red blood cell is taking an exit to enter the vascular network of an organ, where arteries branch into smaller arterioles, passing through subunits of tissues. Finally, you pull into the narrow driveway of the building where you are delivering your package, just as the red blood cell squeezes through a capillary to supply oxygen to a nearby cell. Of course, blood vessels lack street signs. However, we can uniquely identify vessels through a combination of physical properties, such as diameter and branching angle, morphological features, gene expression, and other biomarkers. Altogether, this information forms the Human Reference Atlas VCCF, our roadmap for locating cells in the body.
Thank you for showing that, Bruce. On the next slide, you see how we are trying to get that kind of information. So there's a template that now exists where you have aorta and vena cava and the hepatic portal vein for the helium, and then it goes over to feed those 23 functional tissue units ultimately. So the very first complete or partially complete draft of the Human Reference Atlas might just show you the vasculature and how it connects these 23 functional tissue units, because I think that's all we can do with the current HuBMAP funding, but then there's other funding hopefully in the making. And on the next slide, you then see an example of how that looks for kidney. So if you are a kidney expert, you might like to pour over this and agree or disagree with what you see here. So this really gets to the detailed information on how the blood actually flows through the kidney, and it's complicated. On the next slide, you see the same template completed for the lung. Really not easy to get experts to have so much time with us to get that kind of level of detail, but it's needed in order to truly understand how to build that latitude-longitude system. And just like it took generations to map the map of India, right? It took many generations actually to just go via sidelines across the large, vast land. Here, the human body is also quite large if you are down at the single cell level, and it will take time and much expertise. And we deeply appreciate working with so many experts. On the next slide, you see that there are now also cell type annotations. This is on our end called HRA-POP for HRA cell type populations. And we are using different cell type annotation tools such as Azimuth, CellTypist, PopV, and we are considering adding 2 more to identify cell types. These cell type annotation tools do not always agree when they assign cell types. In fact, in some cases, they have 50% agreement, and all other cells have different names if you run them through these 3 tools. And so we will not be the ones that bring order to this, but we can visualize where these tools agree and where they disagree. And hopefully that helps those which develop cell type annotation tools to improve them. On the next slide, I think that's when Bruce takes over.
Yes. Okay, so we've been talking about the atlas on the core level, like all these things that are gathered by subject matter experts and worked out and hashed out between experts and users. Then the Atlas ++, as in the stuff that's built on top of this core Atlas set of data, is this several different, there are several different things now that we have that core. So the first thing we can talk about is experimental data. We have modeled, so this especially came out of HuBMAP, where we have donors can have, they pull out tissue blocks from the donor, they section it up, they chop it up, and then they run different analyses on it or imaging assays to create these data sets. And the way this data is added or is related to the HRA is via these extraction sites. And what an extraction site does is, so we have this RUI, the registration user interface, which you'll see later, where they say, this tissue block, I extracted it from this part of the kidney. It's very specifically from this part of the kidney. And that becomes an extraction site that's based on these 3D reference organs that you saw earlier. So there's a graphical interface where they say what size the tissue block is and where they came out of. So on the experimental data level, at this first level, they're able to say, okay, this is my tissue block, this is where it came from. And we don't dive into the data. It's just basically cataloging where in the human body, using the common coordinate framework, part of the HRA, did this come from? So this is the registration user interface. Again, so they can put in the tissue block dimensions, and they basically in a web browser, they can move their tissue block to where it came from, make sure that they're happy with it. You see on the bottom right, these anatomical structure tags. Those are Uberon terms. So this will say, this tissue block is colliding with a kidney capsule, cortex of kidney, and other things, so that from that extraction site, we can know, before we even look at the data, these are some of the anatomical structures that may be in that data based on the location, the placement of that data, that tissue block. Let's see. Okay, and so then once we've cataloged all of them, they can be advertised, not just HuBMAP, but they can be federated across multiple consortia. And that's what you see in the exploration user interface. And this, again, is heart, kidney, and lung. We obviously have a lot more data, but for this presentation, we've just focused in on heart, kidney, and lung. So you see those little blocks. Those are all the registrations that have happened as of sometime in June, I think it was. So you can see they can be cataloged together using this experimental data. Oh boy. Yeah, and then the other thing we have this HRA-POP. So this takes the experimental data and then opens up when that data set is a certain type that we can that we support, we can actually open up that data. And that's generally we have both single cell or single nucleus RNA-seq, or there are a few different types of spatial data that we can support, but primarily the ones that can be processed with Azimuth, CellTypist, or PopV. For those, we have a process where we run through those and generate a cell summary. And a cell summary is just, this is the population of cells that I found and their cell types. And the cell types are ontology terms that are associated with the atlas. So that's another way where we get data. We now can say this tissue block has cells from this data set of this type and this many. This is the population of that. So that's what the HRA pop sort of adds. And then we also use that to inform the atlas based on all this. We've actually processed thousands of data sets like this based on the cells that we're finding from these data sets that are associated with these locations in the human body. We can start to say, if you see this anatomical structure based on the data I've seen, this would be the expected set of cells that you might see in that structure. So this is again why this is Atlas++, because it takes experimental data and uses the Atlas core to then create to inform the Atlas further. And here's just some stats about heart, kidney, and lung data sets. See, it was processed. There's 1,000 in this core set that we're talking about here. And then HRA lit is where we take the HRA, so each digital object has papers associated with them. They cite papers that are relevant for that digital object. And in the ASCT plus B tables, each assertion, each anatomical structure, cell type, biomarker pair is given, it should be backed by a specific paper. So we take those papers, we went through to PubMed and found related authors, publications, funded projects, funders. So that then there's a whole nother layer that you can add to the HRA, which is all the academia, all the academic research that's going on around the HRA. And again, so this is from HRA lit version 0.5, about 2.4 million articles for these three organs we identified. Yeah, so this is again, that was the HRA. These are all the pieces of the HRA. And now we can talk about, well, now we have all this cool data, how do we use it? Behind the scenes, each of these types, ASCT plus B table, every table is a digital object, and every 3D reference organ is a digital object. So we process these digital objects to create graphs, an RDF graph that you can query using a graph query language. We use Sparkle, but can be used in other processes. So then each digital object is processed separately and enriched so that by itself, the kidney ASCT plus B table is imminently queryable and useful by itself. But then we combine them together to create the full HRA so that you can, by combining them, you get useful interactions where you can grab the kidney UBRON term, find all the 3D reference organs, the OMAPs, the experimental data associated with it, et cetera. So we publish this as the HRA KG, the HRA knowledge graph, and can be queried, and it was also fair linked open data, so it can very easy to pull these graphs into other graph databases, including Neo4j. So then on top of the HRA KG, we have these APIs so that they can query using Sparkle to create more programmer-friendly interfaces, REST-based interfaces essentially. And then our HRA applications either query the HRA API or query the HRA KG with Sparkle directly. So that's how we sort of build up from our data to user interfaces and applications. So I'll just very quickly, as we're starting to run out of time, just go through some of these interfaces. So the ASCT plus B user interface allows you to create and view ASCT plus B tables. The RUI allows you to register experimental data to the Atlas using these tissue blocks. The exploration user interface has a catalog of all the data that we've registered, and actually the tool itself can be narrowed down for a specific course consortia where needed. And the interactive FTU Explorer allows you to look at the individual functional tissue units and then data associated with it, looking at the cell populations and things like that. Cell distance visualizations allow you to take your spatial data and do some interesting things with it. We're actually working on a paper where we're looking for more data to look at the interplay between cells and the vasculature. And this interface has really simple needs. You just need to say the location and the cell type and in a simple CSV format, and it can be viewed and used. So there's some information there, but we can follow up later. Then the HRA organ gallery in VR allows you to see HRA experimental data and the HRA atlas in virtual reality. And this is one really cool application that you can actually get to in the MetaQuest store. So definitely check that out. And of course, the big where we create an API that allows programmers to then integrate applications at the API level where they can make calls and get reference data, but also experimental data associated with the HRA. And yeah, so we'll go back to user stories because a lot of what we do is driven by the user stories, what the use cases, what people actually need. And so that's why we have such emphasis on these user stories. So,
so if you go to the next slide, you see the table again. And then you have US1, which is to predict cell type populations, which you see here. So you can basically register a tissue block using the registration user interface. And then you get a prediction of how likely it is that there are cells of a certain type based on 3 different cell type annotation tools, Azimuth, Cell Typist, and PopV. So you have to either check out all 3, or you just say, all right, I just want to see predictions by Azimuth of what is typically commonly found in that location. So on the next slide, you see user story 2, which is the reverse. So you have a tissue block which already has a cell type population analysis performed. You know how many cells per cell type you have, and you would like to know where the tissue block might have come from. And we can show you this in a 3D volume, but we can also just show you which tissue blocks are most similar, which anatomical structures are most similar, or which corridors are most similar. On the next slide, you see US3, which is compare reference tissue with aging diseased tissue. So there is no reason why you couldn't also register diseased tissue in here. And that, of course, will have different data behind different cell type populations. And you can now start comparing that. On the next slide, you see that zoomed into the functional tissue unit explorer. And here it's relevant at the functional tissue unit level, not at an anatomical structure or organ level. And that's really relevant for understanding what happens to these functional tissue units as we age, as BMI grows, as disease comes in. And I think there are a number of really interesting applications for that here. On the next slide, you see the cell distance explorer. So again, if you have data that's spatially explicit in 2D or 3D, you can upload that yourself into the CDE browser. And I think Andy might have put that into the chat. So please feel free to check this out. There are also sample data sets that you can explore. It's quite fun to look at the distance distributions from different cell types to vasculature in this case, but you could also look at distance distributions to immune cells, to nervous cells, and others. On the next slide, you see lightweight atlas components in support of sustainability. So these are just HTML code, which you can integrate into your websites, your data portals, to have these human reference atlas data sets show there. And on the next slide, you see US number 7, which is the last but not least a dashboard for the Human Reference Atlas, so that you get to see what coverage and quality it has right now, but also who is using it and what data sets now exist. On the next slide, I think we have it wrapping it up. Bruce, do you want to take it?
Yep. All right. So yeah, so we've given you a very high level overview. I think we've tried to hit every single thing that we have in 30 minutes. So that definitely some follow-up, and happy to follow up with anyone who wants to try things out or have any questions. So for future work, we have releases every 6 months. Every 6 months we add new data, new applications, more improvements, both in June and December. We're always adding more data, more collaborators, more organs. And eventually we want to have the HRA be useful in a clinical setting. And so then we have a very large team, and that has evolved over time, and multiple organizations. And there's some acknowledgements from NIH and NSF, and then some references. References are included, and that's pretty much it. So yeah, feel free to email us, and we have a great website too that has all this information, and happy to take some questions. I know we're running a little short on time, but we can do what we can.
I guess I'll speak for me and hopefully others. That was amazing. I think that the opportunities there are incredible, and I think that it was really cool to see all the different tools. I have a sort of a global question, and maybe others have this question is, I think in many of the consortia right now that are being generated around, a lot of people are focused on bulk RNA-seq or single-cell RNA-seq data in disease state and non from various tissues. And we don't want to have every resource in the universe building their own responses to that. And we also want to capitalize on the work you have done with the HRA, or Human Reference Atlas, which is meant to bring all these together. How do you suggest that somebody like a consortium that is focused on one tissue or one tissue and one disease area, generating a lot of data, how might they best engage with you all? Is it they sort of tour the tools, or is it they just actually have a call with you and say, we have a bunch of data, help us? What's your best strategy from your perspective?
Yeah, so if you, for instance, care about pancreas, you could take the Human Reference Atlas pancreas and just register your data in there spatially. And then that could also be bulk data. It doesn't have to be single cell data. It can be Visium data. It can be other data sets, as long as you know where spatially it came from. And then you can derive cell population information from that, right? Number of cells per cell type, and mean biomarker expression values if you want that in your use cases. And I think that would then still show in the EUI, in our exploration user interface, just as different assay types. And then if people want to run meta-analysis for pancreas, for instance, they would find a lot of single cell data, but then also bulk data and some Visium data and some other data. And currently you have a lot of single cell RNA-seq data and codex data in there, but I think over time we will also have more GeoMx, CosMx, and Visium data in there. Right. But you could also just focus on one organ. There are 35, and people have different types of funding for different organs. And so any combination of existing organs works. We are also currently working on 20 new organs for face, working with dentists. So we also collaborate with teams that didn't find their favorite organ in the existing HRA to add their favorite organs.
Very cool. What about dividing cell types? Because each, in creating integrated maps, how does that work? And can a community that's really focused on the specific tissue help create better maps of the tissues you have? How does that work?
Absolutely, yes. So in many cases, people are forced today to align with cell ontology, but we also know that it doesn't capture everything you really want. It doesn't have cell states and many other things. So we work very closely with the EBI team, which is on our budget actually, and they are very good in adding that in. And so it's just a matter of the community reaching out to us and figuring out how to pay the EBI team because they have full-time staff members which are in production mode. And then to add it. So I think that's a perfect way to harmonize across. And only if all use the same name for the same cell, can you start comparing across technologies and organs and labs.
Are there efforts to is EBI sort of leading the charge for a universal naming or an ontology for all these different tissues? Do you feel, or is that more community driven and you're kind of feeding it, doing it by the communities, or sort of doing it themselves and then collaborating with you all to get this done? What is the...
Yeah, so I think the lung community just did a jamboree and they had a number of really interesting results coming out of that. And there might be other communities which also do their own jamborees. I think it's very organ specific how these societies are organized, what cultures and incentive structures and funding mechanisms exist. And it typically takes a little bit of funding to put up data portals.
Yeah. Well, I don't want to take the mic for everyone's questions, but if anyone have questions, maybe we'll take one before we break. Two. I hear really lots of comments in the chat about this. I love this. I love the 3D thing. I think that's fantastic. I can't wait. We have an Oculus at the house. We're going to try that. Oh, all right.
And then also for training and onboarding, it's great because you get to see everything in 3D and it operates in 3D. We absolutely must embrace 3D, but it's still very expensive to have 3D single cell data. And oftentimes for an entire embryo, it can be done. For the entire human body, it would be massively expensive.
That's incredible. Wow. Mackenzie, any wrap-ups?
Yeah, so I was just going to remind everyone that our next webinar will be in a month on August 30th, and it'll again be from 1 to 2 p.m. We're going to have an introduction of the acute to chronic pain signatures, so please be on a lookout for more details regarding that presentation. But thank you both again, Katie and Bruce, we greatly appreciate it.
Again, everything will be
Thank you.
recorded and on our website, as Mackenzie mentioned. And if you miss it, you can contact us. You can also contact Bruce and Katie to get all the information. But thanks for joining us, everyone.
Thank you.
Thank you.
Take care.
Bye.
Bye.
Bye.